Hello everyone, this is video number 5 for the urinary system, and in this video I will start to cover in more detail the processes involved in physiology of the urinary system. And this slide is from the last video, and this is summarizing the 3 main processes: glomerular filtration, in which fluid moves out of the glomerular capillaries into the capsular space; tubular reabsorption, in which materials move from the lumen of the tubule into surrounding capillaries; and tubular secretion, in which materials move in the opposite direction from the surrounding capillaries into the tubule. This video is going to focus on the first of those 3 processes, glomerular filtration. So a little bit of background, and then we'll get into the details. So glomerular filtration is a passive process. That is, there's no ATP required for this. It's completely driven by a pressure gradient, forcing fluid out of the glomerular capillaries. And we'll get into why that is, what's generating the pressure gradient in a bit. The glomerulus is a really good filter. And it does filter a lot of blood. And remember, in each kidney there are more than 1 million nephrons. So there's a huge number of nephrons containing glomerular capillaries, and that provides a huge amount of surface area. Each glomerulus has a lot of surface area itself. Also, the glomerular capillaries are highly permeable. They're fenestrated capillaries, and if you remember from the vessels chapter, that means they're pretty leaky, so they're pretty permeable. In addition, the glomerular capillaries have a higher blood pressure than other capillary beds in the body, so that's going to be in favor of the pressure gradient as well. Okay, so let's talk about what actually gets filtered during glomerular filtration. We're starting with blood in the capillaries, and some of the material is going to pass out of those capillaries into the capsular space of the glomerular capsule. So what gets across? Mostly what gets across is water and dissolved solutes in the blood plasma. And those are, the smaller the better. So small solutes get across unblocked, I guess you could say. And the larger the solutes are, then there's more difficulty getting across and into the filtrate. So in essence, what happens is a lot of water passes through, and almost all of this, small solutes can get through very easily. But larger substances don't. So the filtrate that collects in the glomerular capsule is mostly containing water with small solutes like glucose, amino acids, ions, and various waste molecules, which are also pretty small. What doesn't get across are the formed elements, white blood cells, red blood cells, and platelets, and the large plasma proteins. They're too big to get across. Now, the structure that is doing the filtering is basically the barrier between the blood and the glomerular capillaries and the capsular space inside the glomerular capsule. And what that membrane is composed of, it's called the filtration membrane, first of all, is the wall of the capillary, which is endothelial cells, and they're fenestrated, so they have those kind of collapsed areas of their plasma membrane. There's the visceral membrane of the glomerular capsule, and if you remember from the last video, those are the podocytes, and the podocytes just wrap around the capillary wall. And then in between them is a basement membrane, which is fused. It's going to be made by both the podocytes and the endothelial cells, and they kind of get glued together. And that is what's referred to as the fused basement membrane here. Okay, so let's take a look at that. And here we are looking at the glomerulus, all the capillaries. Here's the capsular space. So materials are moving from inside the capillary out to the capsular space. Okay, these are the podocytes that are sitting on the surface of the glomerular capillaries. In order to get out, you have to cross this, endothelial cells. The podocytes, they're making up the visceral surface of the glomerular capsule, and the fused basement membrane, which is shown here, right? Fused basement membrane, podocytes, endothelial cells. Okay, so that is the filtration membrane. And this is shown in a little bit higher kind of magnification view representation. And here is the blood inside the capillary. Here are materials that can't get through. They're too large. So white blood cells, red blood cells, platelets, and any large dissolved proteins in the plasma. Here's the endothelial cell membrane. Here is the fused basement membrane. And here are the foot processes, pedicels are called, of the podocytes. These are the filtration slits in which the small water and small dissolved solutes are able to pass through. And so what gets through is now the filtrate. Water plus small dissolved substances. Okay? Now, the process of filtration is driven by a pressure gradient. So let's define these two terms and talk about the pressure gradient and the rate at which filtrate forms. So NFP stands for net filtration pressure. The net filtration pressure is the pressure gradient which is driving filtration of fluid out. Okay? And we'll break down what's contributing to that net filtration pressure. There's some pressure that's pushing fluid out, and that's opposed by other pressures, and the difference between those is the net filtration pressure. Okay, GFR is the glomerular filtration rate, and that is the rate of volume per time that filtrate is forming. So typically, the glomerular filtration rate normally is in the range of 120 to 125 mils of filtrate per minute. So just think about that for a second. Both your kidneys are making, let's say 125 mils per minute. That's an eighth of a liter. In 8 minutes, they're making a liter of filtrate. Okay, so there's a huge volume being generated in both kidneys. In the 2 million or so nephrons that you have in both of your kidneys. All right, so let's look at net filtration pressure first, and that's what's on this slide. Net filtration pressure, there's a net pressure forcing fluid out. We already know that, right? Fluid is moving out. What are the components of the net filtration pressure? So the net filtration pressure is due to, let's look at this equation first, the hydrostatic pressure of the blood in the glomerulus, HP sub G. This is the blood pressure inside the glomerular capillaries. Okay, 60, on average, about 60 millimeters of mercury. Okay, that is opposed by two pressures. One is the colloid osmotic pressure, and if you remember from the vessels chapter, colloid osmotic pressure is due to the fact that there are a lot of plasma proteins in the blood, and there's not going to be very much in the filtrate, right? Because proteins don't exit. So more proteins in the blood, no proteins in the filtrate, and that's going to generate a pressure of water wanting to move by osmosis into the bloodstream. Okay, so that's opposing. There's also an opposing fluid pressure because of the filtrate in the capsular space. That's the hydrostatic pressure of the capsular fluid, HP sub C. Okay, if we do the accounting here, net filtration pressure, the value of the hydrostatic pressure in the glomerular capillaries, 60 millimeters of mercury. Subtract the two opposing pressures, 32 for the colloid osmotic pressure, 18 for the hydrostatic pressure of the capsular fluid. You get a net filtration pressure of 10. So that is the pressure. There's a net pressure pushing fluid out. Net filtration pressure of 10. Okay? Now, one thing you should appreciate about this is the hydrostatic pressure inside the glomerular capillaries is very high as capillaries go. 60 millimeters of mercury, whereas most capillary beds start out at about 35 millimeters of mercury on their arteriole end. The main contributor to this high blood pressure, hydrostatic pressure in these capillaries is the anatomy of the vessel leading in, the afferent arteriole, being larger diameter than the vessel leading out, efferent arteriole. Okay, so there's a superhighway of blood flowing in, and there's a backroad street of blood flowing out. This is going to create a huge traffic jam, and that's greatly contributing to this really high hydrostatic pressure of the blood in these capillaries. That's necessary in order to have a positive net filtration pressure. All right, more about the glomerular filtration rate. So the glomerular filtration rate, we already mentioned the average, 125 mils per minute by your kidneys, both of them. This is directly proportional to the net filtration pressure. Right, that makes sense. The net filtration pressure is driving the process of filtration. If you decrease that pressure, you're going to form less filtrate per unit time than if it stays, at the normal level. Okay, so they're directly proportional. Now, what usually changes the glomerular filtration rate in certain disorders or diseases is changes in blood pressure, which are contributing to changes in the glomerular blood pressure. Just an example, people with chronic hypertension, so have an elevated blood pressure, and that's going to contribute to an elevated blood pressure inside the glomerular capillaries. Chronic hypertension is going to increase the net filtration pressure from plus 10 to maybe plus 12 or 13. That's going to drive an increase in glomerular filtration rate. You're going to filter more blood volume per unit time than you would normally. This is going to generate more fluid flowing into the renal tubules. And that's going to create a problem, a kind of a backlog for the normal process of reabsorption. Most of the fluid that gets filtered gets reabsorbed. But if you generate a much higher volume, then that's going to cause a problem with reabsorption. As a result, not all of the fluid that's normally reabsorbed would be, and more of this will pass into the urine. And so more of the normal things that your body needs and reclaims will instead be lost in the urine. So that's one of the complications of chronic hypertension, is what it's doing to your kidneys in terms of the process of blood filtration. Okay, I'm going to stop this video here, and in the next video I'm going to talk about the other two processes: reabsorption, tubular reabsorption, and tubular secretion. See ya.